So I believe we finished last week a conversation. We finished talking about stomach, right? And now we're moving towards the duodenum. So duodenum is a fairly short fragment of the small intestine. Don't get tricked by its size. It's probably the most important digestive organ in the entire alimentary tract. It's about an inch in diameter, about 10 inches long. Now, it is connected to two secondary digestive organs, actually three secondary digestive organs: liver, gallbladder, and the pancreas. Well, these three organs release their secretions into the duodenum by the structure called hepatopancreatic ampulla. Let me choose the different color. So this here is hepatopancreatic ampulla. At hepatopancreatic ampulla, two ducts, main pancreatic duct and main bile duct, merge together, releasing the combination of bile and pancreatic juice into the duodenum. Duodenum is mainly controlled by parasympathetic system through vagus nerve. Sympathetic innervation is through thoracic splanchnic nerves. Superior mesenteric artery delivers the blood to the duodenum, and the activities that this organ carries out are essentially main portion of the chemical digestion of the food. And a lot of nutrients are getting absorbed in the duodenum. Therefore, for effective absorption as well as secretion, duodenum requires an increased surface area. And that increased surface area is achieved by three surface modifications that can be found in the duodenum. First surface modification are circular folds. Can you see those? Those circular folds are essentially folds that go circumferentially around the duodenum. So basically it's like, they're like waves. They're very noticeable with the naked eye. They're about half an inch, maybe a bit less tall. And they are not stretchable. The duodenum is not supposed to stretch out, and these folds are not supposed to be smoothed out like rugae in the stomach. Okay? One of the reasons why the importance of circular folds, not only to increase surface area, they also allow the chyme that comes from the stomach, that portion right here, they allow that chyme to start circling over the surface of the duodenum, which slows the chyme down and increases the time for absorption. Does that make sense? Second surface modification that can be found in the duodenum are villi. This finger-like projections, about 1 millimeter high, give duodenum a velvety texture. If you will open it up, okay, and look at it, it's going to look smooth and somewhat velvety. Villi can be seen by the naked eye. Okay? Does that make sense? Actually, presence or absence of villi is a really good sign of the sort of health of the intestine. In the disease known as inflammatory bowel disease, villi are largely gone, and the surface of the inflamed ileum, for instance, looks like cobblestones. This is the sign of the abnormality in the intestines. Lack of villi in the diseases like IBS, inflammatory bowel syndrome, contributes to the diarrhea and abdominal discomfort that patients with IBS experience. Inside of the villi, you can see the model right here. That's a villus holding a villi. Inside of the villi, you can find blood vessels, as shown here in red and blue, and lymphatic vessels that are called lacteal. So capillaries, venules, these blood vessels in the villus, they absorb carbohydrates and amino acids, while lacteal is responsible for the absorption of fat. Close to villi are mucosa-associated lymphoid tissue. It's located in the lamina propria of the mucosal layer of the intestinal wall and responsible for protection against the pathogens that come with the food. Now finally, the third surface modification that dramatically increases the surface area of the duodenum are microvilli. So in this image, you can see a border, so-called brush border, on the apical surface of the columnar, simple columnar epithelium that lines mucosa of the intestine. Microvilli are essentially the folds of the cell membrane that additionally increase the absorptive surface in the duodenum. So far we have three structures: circular folds, villi, and microvilli that increase the absorptive surface in the duodenum. Does that make sense? Another function of brush border microvilli, or as we call it, a brush border, is to harbor the enzymes, so-called brush border enzymes, that are necessary for a complete digestion of carbohydrates and proteins. Now you can see that there are crevices, crypts, in the epithelium of the small intestine. These crypts are called intestinal glands. These intestinal glands in the duodenum harbor four major types of simple columnar cells. Absorptive cells, with the layer of brush border layer of microvilli that responsible for absorption of nutrients. Goblet cells here that produce alkaline mucus. So pH in the small intestine is slightly alkaline, which makes perfect sense because the chyme that comes from the stomach is acidic, right? So in this case, alkaline mucus will neutralize the acidity of the chyme. Enteroendocrine cells, which produce two main intestinal hormones, cholecystokinin and secretin. And finally, Paneth cells. Paneth cells produce protein, antibacterial protein, called lysozyme, that can destroy bacteria coming with the food. They may also work as phagocytes, effectively engulfing and destroying pathogens that contaminate the food. Very close to the junction between the stomach and the duodenum is Brunner's gland, submucosal Bronner's gland produces thick alkaline mucus, which again makes perfect sense considering the acidity of the chyme that comes from the stomach. So to summarize what we just learned about the mucosal surface of the duodenum, it's the simple columnar epithelium that has a lot of intestinal crypts or glands. There are 4 major cell types: the absorptive cell, the goblet cell, endocrine cell, and Paneth cell. Absorptive cell has brush border on its surface, which harbors enzymes necessary for digestion and absorbs nutrients. Goblet cell produces mucus. Endocrine cell produces intestinal hormones. And Paneth cells protect against the microorganisms by releasing antibacterial lysozyme and phagocytosing pathogens. We're good? Any questions before we move forward? Now let's talk about brush border and its enzyme. The picture on the right shows you the brush border. You see microvilli, the membranous folds, right? Covered with a layer of mucus. Not only this, these microvilli have proteins called brush border enzymes embedded in the membrane of microvilli. So what do these enzymes do? They complete the digestion of nutrients before elementary building blocks are absorbed. Now, what do you need to know about these enzymes? As you can imagine, I'm not going to ask you to provide me with the list of the enzymes. We have multiple choice, not short answer questions. But for each enzyme in this table, you must know the substrate and the product of the reaction that this enzyme facilitates. So we're going to start with the enzymes that break down carbohydrates. Dextrinase breaks down small oligosaccharides called dextrins, and dextrins in turn are the products of the starch breakdown. We're clear? So dextrinase breaks down dextrins into individual molecules of monosaccharide glucose. Maltase breaks down disaccharide maltose, which is also a product of the starch breakdown, the glucose as well. Sucrase breaks sucrose, not really, surprising, to 2 individual monosaccharides, glucose and fructose. Finally, lactase breaks down milk sugar, lactose, to glucose and galactose. Does that make sense? Can you remind me in which other part of alimentary tract carbohydrates are digested? Mouth. Yes, correct. Now this enzyme, enterokinase, kind of interesting. Enterokinase does not break down anything. The function of enterokinase is to activate the protein called trypsinogen, and the result of that activation is the protein called trypsin, one of the key enzymes responsible for the digestion of proteins in the intestines. Now I want to clarify, trypsinogen doesn't come from the intestine. Trypsinogen is the enzyme produced by your pancreas. So far. Good? These two enzymes, aminopeptidase and dipeptidase, break down peptides. Aminopeptidase chops off the amino acid at the amino end of short peptides. Every peptide, say this is the peptide, okay? 5 amino acids, okay? Every peptide has the amino end and carboxyl end. So aminopeptidase will be chopping off peptides, will be chopping off amino acids from the left. Does that make sense? One by one. One by one. Does it make sense? Like unlinking the cars in the freight train from the end, one by one. Does that make sense? Dipeptidase breaks down dipeptides. So substrate for dipeptidase is the molecule that consists of 2 amino acids. And it will break it down and you will have 2 amino acids individual. So far, you clear? Yes. What is the substrate of aminopeptidase? You get me a list. What is the product of sucrase? You get a list. Make sense? It can be someone. What is the function of dipeptidase? Break down dipeptides into amino acids. Make sense? Now, enzymes called nucleosidases and phosphatases here, they break down nucleotides, elementary building blocks of RNA and DNA, into individual parts, nitrogenous bases, pentose sugars, and phosphates. Does that make sense? So this is pretty straightforward, as Marina asked, that's what I'm going to ask. What is the function of maltase? Break down oligosaccharides into the glucose. Any questions? Moving on. So, what is the role for these two enzymes, these two hormones that I mentioned before? Secretin and cholecystokinin. So let's take a look at the whole story about the regulation of digestive activities in the intestine. Let's start. Remember, it's all about reflex and it's all about negative feedback, right? So the first thing that happens in the intestine is that chyme enters the intestinal lumen from the stomach. Does that make sense? What is the pH of the chyme that leaves the stomach? Acidic. I rarely look for the exact number. You're right, it's acidic. So acid in the duodenum, acid shouldn't be there, right? Acid is not supposed to be in the duodenum. Acid will stimulate the release of secretin. Along with acid, there are a lot of other chemicals like proteins, amino acids, peptides, oligosaccharides. And what's important, there are fats. So fats induce cholecystokinin release. Does that make sense? This image, you see cholecystokinin being red and secretin being yellow. Okay? So remind me again, which stimulus leads to the release of secretin? Acid, right? In order to establish the negative feedback here, what should happen to the acid? It should be neutralized. And what neutralizes acid better than the base? So secretin will stimulate the release of bicarbonate by the pancreas. Okay. Now, what should be a response to the presence of fats? What should you do with fats? Break them down. What breaks fats down? Enzymes. Cholecystokinin stimulates the release of pancreatic enzymes from the pancreas. Does that make sense? So the effects of secretin on the pancreas is that it stimulates bicarbonate release. Effects of cholecystokinin on the pancreas is that it stimulates the release of enzymes. Are we clear so far? We good? What about liver? Turns out secretin, to some extent, stimulates the release of bile from the liver. Liver starts producing bile. Does that make sense? But what is probably more important effect is that cholecystokinin right here stimulates the gallbladder. Gallbladder normally stores bile, and cholecystokinin stimulates gallbladder to release the bile. Sorry, I missed it. Stimulates gallbladder to release the bile into the duodenum. Does that make sense? So, bile gets in the duodenum. The function of bile is to make lipids more soluble. Okay? Now, let's wrap it up again. Food, acidic chyme, enters the duodenum, right? Acid in the chyme stimulates what? Secretin. Secretin stimulates the release of bicarbonate from the pancreas and, to a lesser extent, stimulates the release of bile from the liver. Lipids in chyme stimulate which hormone? Cholecystokinin, which stimulates the release of enzymes from the pancreas and contraction of gallbladder and release of bile from the gallbladder in the duodenum. It turns out when bile gets in the duodenum, it contains so-called bile salts. These bile salts will go back into the circulation and they will stimulate the release of bile from the liver. Bile will enter duodenum. Bile salts will get in the circulation, will stimulate the release of bile from the liver. As long as there is chyme coming from the stomach, bile salts will perpetuate that bile release from the liver. Does that make sense? Bile salt release is actually a positive feedback loop. Does that make sense so far? So basically, we have two main organs, secondary digestive organs, that are responsible for chemical digestion in the intestine. One is the pancreas, that produces bicarbonate and pancreatic juices. And second is the liver, that produces bile. Well, gallbladder is also kind of important, but it doesn't produce anything, as we will see soon. So far, is that clear? Another thing that happens in the intestine, in the duodenum, is segmentation. Okay, segmentation happens in response to these two hormones, cholecystokinin and secretin, and it ensures that chyme is mixed better with all the juices, pancreatic juices, intestinal secretions that are present in the lumen of the intestine. Peristalsis, pushes the bolus of food further towards, other parts of the small intestine for absorption, so on and so forth. Now you can see that I'm not going to ask numbers, but I mentioned that food stays in the stomach for about 2 hours. Food stays in the duodenum for up to 5 hours, because that's where the most of the digestion and absorption happens. Understand? It stays there longer because that's where digestive activities happen. Any questions on this? Yes? About 2. I'm not going to ask the numbers. Basically, in duodenum, it's much longer. So, this is what. So, we have to consider duodenum, liver, and pancreas kind of in complex. And we're going to move on to talk about liver. It's the largest internal organ, okay? And second largest overall organ. What's the biggest organ, largest organ in the human body? Skin. Yes, by mass, by volume, by surface. Liver weighs from 2 to 4 pounds, okay? Represents about 2% of the body weight. It is located below the diaphragm in the right upper quadrant of the abdominal cavity. It receives blood supply from hepatic artery. Hepatic vein, on the other hand, goes in a really interesting and fascinating way. Hepatic vein brings in the blood from both GI tract, spleen, and pancreas. And hepatic vein represents what we call a enterohepatic circulation. Now, we will talk about the function of the liver, but think about this. Liver, here, is kind of connected to the intestines so that blood goes from the intestines to the liver. Does it make sense to you? If it doesn't, I will show you in the minute how this enterohepatic circulation works out. So anatomically, the liver consists of 4 lobes. I expect you to know all 4 of them. The biggest, right lobe. Second biggest, left lobe. Okay, divided by the falciform ligament right here. Smaller caudati and quadrati lobes of the liver can be found on its posterior surface. Okay. Liver is suspended to the diaphragm by the ligaments. In terms of liver anatomy, you need to know this 4 lobes of the liver.
Can you repeat that one more time?
Absolutely. So, first of all, image-like, so this one is anterior view. You look at the liver, imagine that somebody got the abdominal cavity completely exposed. This is what you would look at. This is the posterior view. So that's posterior surface of the liver. Make sense? Right, left, caudati, quadrati. Better? Yes? So, let me... The caudati lobe, quadrati lobe. Sorry. All right? Moving on. So, functionally, liver consists of multiple liver lobules, like this. Okay? So each liver lobule is basically an independent functional unit of the liver. So what do these lobules consist of? They're made of the plates the liver cells. That's a plate. It's another plate. These plates are called plates are made of hepatocytes. Sorry, liver cells. Okay, you can see that these plates are penetrated by a bunch of capillaries called sinusoids. So these blue lines are capillaries called sinusoids. Sinusoids are extremely permeable capillaries. So proteins, carbohydrates, all nutrients can fairly easily go back and forth between the blood and the hepatocytes. Am I clear? So here, in the corners of the lobules, you can see the structures that are called liver triads. In the portal triad, or liver triad, you can see the portal arteriole, the portal vein, and the bile ductal. So portal arteriole brings in the arterial blood from the hepatic artery. Portal venule, or portal vein right here, brings in the blood that comes from the digestive system. So blood that goes in here. Can you see the arrow that I'm drawing? Can you see it? So that arrow shows you the direction of blood. Blood goes from intestines. So blood that goes through the portal vein is enriched with nutrients and everything that gets absorbed in the intestines. Does that make sense? So it reaches liver through the portal vein, and then it is mixed with arterial blood from the portal arteriole in the sinusoids. So what happens in the sinusoids? Liver receives oxygen. It also receives all the nutrients that come from the digestive system. And liver modifies them, stores them, repackages them, It's responsible for a lot of metabolic processes, as we will see in a minute. So far, does that make sense? Blood from sinusoids ends up in the central vein, located at the center of each liver lobule. All central veins, they merge together, forming a hepatic vein that leaves liver and enters vena cava eventually. Okay? Now, I want to trace the pathway for the nutrients from the digestive system. So we're going to start with the intestines. From intestines, these nutrients, where do they go? To the portal vein. From portal vein, they reach into, liver sinusoids. From liver sinusoids, they will get into central vein. From central vein, they will get into hepatic vein. From hepatic vein, they will enter vena cava. And from vena cava, they will enter... We didn't cover that. Anybody knows what where does vena cava bring blood to? Heart. And heart will distribute that blood all over the place. Does that make sense? So basically, you can see that nutrients that were absorbed in the duodenum, they go through the liver for repackaging and modification and cleanup and stuff like that. Does that make sense? Okay? Now you can imagine that blood that comes into the liver from digestive system can carry microorganisms or crap or toxins, special cells called reticular endothelial cells or macrophages or Kupffer cells. They essentially clean up the blood. They consume all the debris and other stuff. Does that make sense? Now, so far, we figured out the function of the portal arteriole and the function of the portal venule. And look at the blood flow. So on this picture, using directional, like from bottom to top, from top to bottom, on this picture, the blood in the portal arterioles and portal venules, in which direction does it flow? From up, from bottom to top, right? Now, what about the ductal here? The bile ductal. It turns out that hepatocytes, in addition to metabolic functions, also produce bile. And this bile, through bile canaliculi, enters the bile ductal and then drains towards the bile duct and eventually ends up in the duodenum. Does that make sense? So to kind of wrap it up, you got the sequence. I don't want to go through the sequence anymore, but you got the sequence, wrote it down. So this is how blood flows through the liver. Sinusoids allow the exchange between the blood and hepatocytes. Liver can absorb nutrients. Liver can secrete some, can release some molecules like glucose or lipoproteins or fats into the blood. And then all these nutrients go into systemic circulation eventually. Another function of hepatocytes is to produce bile, which is drained into the bile ductals through the bile canaliculi. Okay, and eventually ends up in the duodenum. So far, we're good? This is what are the components of bile. First component, bicarbonate, the base. It neutralizes acid. Remember, bile goes into the intestine. Bile goes into the duodenum. So, why there is a need for acid neutralization? Acid comes from the stomach, exactly. So, that's number one, okay? Second thing, emulsification of lipids and phospholipids. Now we need to figure out the word emulsification. Imagine that you have a skillet with oil on it. Not the fat, the oil. Oils, are they water soluble? They aren't. Now imagine you take this skillet with oil and you pour some water in it. What you gonna see? Huh? Separation. Yeah, they're gonna be like oil chunks and bubbles floating in the water. You with me so far? You take a bit of the dishwasher gel or liquid, whatever, and you add this bit into that mixture of oil and water. What's going to happen? Huh? They're going to mix. They're going to form emulsion. So these huge chunks of oil are going to start breaking apart, and you will see it. Like they break apart and they form maybe even almost invisible, tiny little bubbles that are called micelles. Okay, we're going to talk about micelles in a minute. But basically what you did with that dishwasher gel, you emulsified the lipids. You turned them into the emulsion. Definition of the emulsion, it's called colloidal solution. So it's not a real solution, right? But emulsion is a colloidal solution of oil in the water. That makes sense? Milk you buy milk. How does it look? It's white, right? Homogeneous, white homogeneous fluid. Okay? Say you buy farmer's milk, non-pasteurized, but still, it's white homogeneous fluid. You put it in the can, you stick it in the fridge, you take it out a day later. What's going to happen to the milk? Separate into cream, well, and the rest of it. Okay? So, milk is an emulsion of lipids in water. Does that make sense? When you leave it standing, lipids float on the top. So emulsion eventually will separate. Does that make sense? Bile salts and lecithin are necessary for that emulsification. Another little explanation why emulsification is important. Did you ever read the composition of chocolate? Does it contain oil? Oh, yes, it does. Cocoa oil. Okay? And the more it has it, the better the chocolate. Now, as you can imagine, the rest of the chocolate's kind of solid, so if you just dump the oil there, you're going to have a giant blob in the center of your chocolate bar, and it's not going to be really tasty, right? So in order to emulsify that cocoa oil when you make chocolate, people add, well, people, companies, food producers, add lecithin into it. Don't worry, lecithin that goes into your chocolate doesn't come from human bile, no. It comes from plants, okay? But lecithin is necessary to emulsify that cocoa butter, cocoa oil that goes into the chocolate. Does that make sense? Bile pigments. They are actually products of the degradation of heme. Cholesterol. Cholesterol has to be excreted. Cholesterol and cholesterol-derived hormones. And bile contains mucin. Remember that protein that is necessary to form a good bolus of food. Mucin allows food particles in your intestine to stick together better. Now let's talk about some of the bile components a little bit more. So first, bile acids. So this part is cholesterol, okay? This is acid part. So essentially, this molecule of bile salts represent what we know as amphiphilic molecules, okay? Amphiphilic means that they have non-polar part. That's non-polar part. And they have polar part, this one. Okay? So how does bile emulsify lipids? Non-polar parts will bind to the lipid blob, while polar parts will stick out. Does that make sense so far? So the outer surface of this structure will be what? Hydrophobic or hydrophilic? Hydrophilic. Yes. Those black ones are the hydrophilic parts. While the inner surface will be hydrophobic. That's where the lipids are. These structures are called micelles. Does that make sense? The whole thing. Like, so micelle is the structure which has lipids in the center, and these lipids are basically emulsified by the bile salts. The same way you emulsify oil when you wash the skillet with a dish soap. Does that make sense? Because otherwise, all fat in your intestine, and your intestine is really watery, right? There's water secretion. So all the fat in your intestine will just form a giant blob. Okay? That makes sense? Now another important component of bile that I want to focus your attention at is bilirubin. It's actually a great story. So red blood cells normally degrade. They degrade in the spleen. That's normal. They last for about 3-4 months. 4 months they expire. Red blood cells get degraded, and as the part of the degradation, the chemical known as heme, it's a part of hemoglobin, okay, is released in the blood and delivered to the liver. We clear? Now, heme is pretty toxic, and we need to convert it. And heme is converted through the step with biliverdin. It's converted to bilirubin. Does that make sense? Now, bilirubin becomes a part of bile and is released into the intestines. And bilirubin is what gives intestines the brown color. Not intestines, sorry, feces. Bilirubin is responsible for brown color of feces. Sort of a kind of real-life clinical connection. People that experience, for instance, some liver abnormality, will get to jaundice. Jaundice is induced by increased bilirubin. We'll get to this. That's a good point. But sometimes people abuse alcohol one evening, a lot. And liver takes a hit. And at some point, liver will stop properly functioning. And it will stop producing normally colored bile. And in this case, especially after really severe alcohol consumption in the morning, what you're going to see is the color of feces is going to be lighter than usual. If it was really a lot of alcohol consumed the night before, it can go to gray instead of brown, because liver just loses its function temporarily. Does that make sense? Now, bilirubin gets in the feces. Some of it will leave the organism with feces, but some of it will be converted from bilirubin into the compound called urobilinogen. Now, urobilinogen will go back into the circulation and will be excreted by the kidneys. And this is what gives the kidneys the amber color. Does that make sense? Yes. Sure. So bilirubin is excreted from the liver with the bile. Goes in the intestines. Some chunk of excreted bilirubin will color feces brown and will be excreted with feces. And another chunk will be converted by intestinal bacteria into the compound known as urobilinogen. Urobilinogen will be absorbed in the blood, in the intestine, will get to the kidneys, and will be excreted through the kidneys, giving urine its amber color. Does that make sense? Now, this is intrahepatic circulation. Look here. So you've got gallbladder, and you've got liver, and you've got bile duct. So bile duct will release bile into the lumen of intestines. Does that make sense? Turns out bile salts will be reabsorbed by a hepatic portal vein and will reach liver back. Remember, I told you before that bile salts can stimulate production of liver. That's your cycle. So bile salts will get through the bile duct into the intestine, will be reabsorbed through the portal vein, reach the liver, will stimulate bile production. Bile contains bile salts. More bile salts will enter the intestine. More bile salts will get reabsorbed in the portal circulation, and the cycle will be perpetuated as long as you have absorption and you have food in the intestine. Does that make sense? Another important aspect of intrahepatic circulation is that there are, for instance, different drugs that can be reabsorbed. For instance, some drugs like ampicillin or streptomycin, tetracycline are highly effectively reabsorbed. So they circle through this a lot. Because liver is responsible for detoxifying. Does that make sense? It will excrete drugs normally. Think about this. If you give a patient tetracycline, you can be sure that tetracycline will stay in the circulation for a while because it's very effectively reabsorbed in the duodenum. But if you give patients sulfonamides or penicillin, penicillin isn't easily reabsorbed. Does that make sense? And it will be excreted rapidly with feces. Am I clear? So when doctors, when farm companies, they look at the pharmacological kinetics of the drug, they look at basically the rates of reabsorption of the drug in the intestine. If the rate is high, if drug is effectively reabsorbed in the intestine, it's going to stay in the circulation for a while, and you can give lower doses. Make sense? If it is very effectively excreted, then your poo is going to smell with penicillin. You will have to take it more frequently in the large doses. Does that make sense? Now, what are the functions and what are the problems with the liver? Functions. First, as we mentioned, detoxification of the blood. Okay? So liver excretes various molecules in the bile, contains Kupffer cells that phagocytose pathogens, chemically modifies drugs. Okay? Actually, when drugs are tested, they are often tested in vitro using a liver extract to see if the drug composition and drug function is affected by liver enzymes. And they modify toxic products of human metabolism to less toxic. So liver will produce urea and uric acid, which are the less toxic products of the protein catabolism. Liver is responsible for carbohydrate and lipid breakdown and storage. Lipid will convert, say, fructose to the glucose. Okay? Liver will store glucose in the form of glycogen or fat. Does that make sense? So I'm kind of jumping a little bit ahead of a train, but if you eat food that contains a lot of carbohydrates, first what you're going to use these carbohydrates for is energy, correct? Then at some point when you don't need any more energy, when all ATP capacity is being completely fulfilled, carbohydrates will be stored in the form of glycogen, right? When glycogen storage is filled, carbohydrates will be stored in the form of fat. The majority of that fat will be directed into the adipose tissue. But if you really eat a lot, like a lot, some of that fat will be stored in the liver, leading to so-called non-alcoholic fatty liver disease, NAFLD. Very common disease in people who suffer from the morbid obesity. Liver is responsible for lipid metabolism, not only cholesterol excretion, and we'll see how important it is, but also for instance, repackaging of triglycerides and sending them towards the adipose tissue. And when you decide to go onto the keto diet, liver is responsible to produce those ketone bodies from fatty acids that will keep you running in the absence of carbohydrates. Liver produces a lot of proteins. Main protein of the blood, albumin, is produced in the liver. Beta globulins, the plasma proteins that transport hormones and other fat-soluble molecules, produced in the liver as well. Finally, liver is essential for clotting factors. Okay? And of course, bile. Now you see how many functions liver carries out. How long do you think a person can live without kidneys? Both of them. Huh?
Not very long.
Give me a number. Minutes, hours, days. Days. Some can last up to 2 weeks. It's going to be miserable 2 weeks, but days. How long without liver? 18 about 18 hours. If liver fails, we're talking about less than a day. Because, yes,
How can the liver regenerate?
it can regrow. If not, it's not completely destroyed, it can regrow. So, for instance, it's actually a pretty good, so when we say regenerate, we essentially mean, for instance, somebody has liver cancer. Okay? Surgeon can remove the entire lobe to make sure that all cells, all cancer cells are gone, and it will grow back. Okay? Yes?
Somebody with a bad kidney, they go to dialysis. What is that?
Dialysis is artificial kidney, essentially.
Oh, no.
Kidney normalizes the concentrations. Dialysis normalizes the concentrations of, ions and pH. Now, the main liver issue, cirrhosis. The mechanism. So first of all, we tend to think that cirrhosis, liver cirrhosis, affects people who abuse alcohol. That is not true. Well, it does, but not only that. Cirrhosis is the result of liver inflammation. This is a cirrhotic liver from dog. Okay, couldn't find good image of human cirrhotic liver, but it's going to be looking just the same. So what exactly happens? When liver is damaged, there is an inflammation, correct? When there is inflammation, there is repair. And how is something repaired? Scar tissue. Scar tissue consists of what type of tissue? Mainly connective. Connective tissue is not hepatocytes. So as scar tissue accumulates, liver loses its function. Does that make sense? And what happens when liver loses its function? First of all, it cannot chemically alter toxins and drugs. Toxins will start to accumulate in the blood. Okay? It cannot produce clotting factors, blood clot. It will fail to excrete steroid hormones. It's very noticeable in men with cirrhosis. They develop a condition known as gynecomastia. Men normally produce estrogen in small amounts, which is completely excreted by the liver into the body. When liver fails, estrogen starts to build up in the blood, leading to feminization of a male. And the first site is gynecomastia, and as one of my former students put it when we discussed it, she said, "Oh, now I understand why those folks grow tits. They essentially grow breasts, female breasts, okay? They start developing female breasts. Not only that, people with failing liver will develop various fluid balance abnormalities, basically edema, because albumin, this protein here, is the key element in the maintenance of fluid balance between the blood and the tissue. Failure to produce albumin will lead to the peripheral edema, swelling, and so on and so forth. Does that make sense? Well, as I mentioned, alcohol consumption is the cause for cirrhosis. Diet. Turns out consumption of large amounts of high fructose corn syrup is directly linked to the non-alcoholic fatty liver disease. Infections, mainly viral infections, hepatitis C, hepatitis B, will lead to cirrhosis eventually. But it can be parasitic infections, not necessarily this. Does that make sense so far? Gallbladder. It's pretty boring. It's a sac that stores bile. So bile salts will go into the intrahepatic circulation, and you have positive feedback that regulates the production of bile. Okay? So at some point, when all the food, all the chem in the intestine is digested, all the excess bile will be stored in the gallbladder. Gallbladder will concentrate the bile. And now think about this. When chem, say, you're done with your breakfast, you digested it, you're doing lunch, okay? So eventually chem will enter your duodenum. That will lead to the release of two hormones, cholecystokinin and secretin. And cholecystokinin specifically, what does it do to gallbladder? Stimulates bile release. Yeah, so bile can emulsify lipids that you have in your meal. Does that make sense? So gallbladder releases it. In the intestine, there's 3 layers: mucus layer, muscularis, serosa. See, there's no submucosa because there's no absorption. Okay? Now, you need to know the pathway of the bile as it is released from the liver and gallbladder. So this is liver here. Liver produces bile, which is collected into 2 ducts from right and left lobes of the liver, right hepatic and left hepatic duct. They merge and form common hepatic duct. Common hepatic duct merges with the cystic duct that comes from the gallbladder, forming common bile duct. Common bile duct joins pancreatic duct at the hepatopancreatic ampulla. Okay, so you can see here pancreatic duct in the center and common bile duct. They merge at the hepatopancreatic ampulla, releasing the mixture of the blood and pancreatic secretions into the duodenum. That makes sense? Yes.
What happens after the right and left merge form the common one?
What do you mean by what happens?
Like, what's the step that happens after that?
It's just structural. Let me do one thing, and if it's still unclear, we'll try something else. Okay? Look at the right picture. So you have right and left ducts. Can you see those? What's in those ducts? Bile. I'm not asking any hard questions. So they merge and they form common hepatic. Still bile. And common hepatic merges with cystic. Still bile. They form the common bile duct. It's still bile. Does that make sense? Am I answering your question? Okay, good. And then finally, common bile and the pancreatic duct will merge. Secretions will be mixed, will be released in the duodenum. Does that make sense? The problem is when bile gets concentrated, it produces gallstones. Gallstones consist mainly of cholesterol, some calcium, some bile salts. Risk factors being white, being obese, being inactive. Okay? Producing a lot of estrogen, being older. Doesn't mean that it affects only older obese women. It can affect basically anybody. When gallstones block the cystic duct, gallbladder starts to swell and getting inflamed, a condition known as cholecystitis. Okay? Treatment now is just cholecystectomy. So basically, it's really, really painful in the right upper hypochondriac, right hypochondriac region, not upper, under your ribs here. Okay? People go in the emergency room, get an operating table, gallbladder is removed. It's really fast. It's a day or two, I think, in the hospital. It's really, really quick operation. Bile is still produced, but you don't have that storage. So you have to be really careful eating fatty food. Because if you eat a lot of fat, it takes time for liver to get up to speed producing bile. That make sense? So there is a world record. The largest gallstone ever removed from a person, so that person was still alive afterwards. Size, any guesses? Huh? Cannot hear. Golf ball. I prefer weight. Guesses? 8 ounces. What else? Half a pound. Okay. Huh? 10 pounds? Getting closer. 14 pounds. So 14 pounds gallstone. Just one. That was one. That wasn't like total mass. It was just one gallstone. Okay. It was a woman in England. She was obese, yeah. But that's pretty remarkable. Okay. Huh? They pretty light, so it should be fairly large. It's probably like a melon. Yeah. Well, of course, you know, they removed gallbladder as well. But she survived. Yeah, it's a mass of a baby. Yeah. Well, and it's a pretty large baby, if you ask me. Mine were smaller when they were born. So, say again?
How do you let it get that bad? That sounds painful. How do you wait that long? Yeah.
We actually, since pancreas takes a lot of time to talk about, let me see. So you know what? We're going to chat about pancreas. We're going to take a break, and I will answer your rhetorical question about how people can do it. So, pancreas, retroperitoneal organ. You can kind of see it here. So this is a pretty good picture actually on the left. So that's a picture of cadaver. You can see a liver, partially resected. You can see gallbladder. You can see pancreas, and you can see the dissected duodenum. Okay? So pancreas has 3 loosely defined parts: the head, the body, the tail. Tail is proximal to spleen. Okay? Does that make sense? So it spans basically from duodenum to spleen. And we know about endocrine functions of pancreas, but we're not interested in those here. Exocrine functions are performed by acini. So you can see acinar cells. Here you can see the cross-section of the pancreatic duct. These acinar cells, they produce pancreatic juices. Okay? So cell releases pancreatic juices into the lumen of the duct, and it flows, eventually ending up in the duodenum. Does that make sense? The whole idea. Okay? The cini produce pancreatic juices. Pancreatic juices end up in the duodenum. So what do they contain? First and foremost, they contain bicarbonate. Look at the picture on the right. You've seen it before, have you? Remember we talked about stomach? How carbon dioxide diffuses into the cell, interacts with water, and forms carbonic acid. And then carbonic acid dissociates into bicarbonate and hydrogen ions. But in case of pancreas, the part that you need is bicarbonate. Does that make sense? So bicarbonate that is formed in the acinar cells is released into the lumen of the pancreatic duct, while hydrogen is returned into the blood. Am I clear? I want you to focus on this process. I'm not going to ask you, I promise, I'm not going to ask you on the transport of potassium or chloride, okay? Not my focus. Am I clear? I want you to focus on what happens in the cell. CO2 interacts with water, forms carbonic acid. Carbonic acid dissociates to bicarbonate and hydrogen ions. Am I clear? We good? Make sense? What else is in the juices? Enzymes. That is a lot. As I always say, how do you eat an elephant? Piece by piece. So you gotta put them in the groups and study by groups. First, 4 enzymes responsible for the breakdown of proteins. Okay? So first enzyme is trypsin. Trypsin is derived from the precursor called trypsinogen. Trypsinogen gets activated by enterokinase. Becomes trypsin. Trypsin breaks down proteins to peptides. Does that make sense? Basically, trypsin breaks down proteins. Those are proteases. So far, we're clear? Now, trypsin activates a bunch of other enzymes. So trypsin will activate chymotrypsinogen into chymotrypsin. It will activate proelastase into elastase. It will activate procarboxypeptidase into carboxypeptidase. It will activate prophospholipase into phospholipase. Does that make sense? So, one more time. Enterokinase, which is the brush border protein, activates trypsinogen so it becomes trypsin. And then trypsin is responsible for activating chymotrypsinogen, proelastase, procarboxypeptidase, and prophospholipase. So this is all be facilitated by trypsin. Does that make sense? Now functionally, first 4, what you need to know about this. They break down proteins. They are proteases. And the product of that breakdown are peptides and amino acids. Now, phospholipase, what does it break? Phospholipase Phospholipase. Phospholipids. Yes. To phosphate glycerol and fatty acids. Lipases. What do they break down? Fat, lipids. So separate fatty acids and glycerol. Amylase. You've heard that name before. Huh? Carbs. Cholesterol esterase. If cholesterol is bound to something, cholesterol esterase will separate cholesterol from something. Ribo and deoxyribonucleases break which molecules? RNA and DNA. Ribonuclease breaks ribonucleic acid. DNA breaks deoxyribonucleic acid. So, type of question. Trypsin breaks down fats, breaks down carbs, breaks down proteins, breaks down nucleic acid. Proteins. Chymotrypsin, produced directly in pancreas, or another version, has to be activated by trypsin. Has to be activated by trypsin. Does that make sense? You will not have to produce a full list for me. But I do demand knowing the function of each enzyme and which ones require activation. Am I clear? We're good? Let's take a break.